Ebolavirus has a 50 to 90% fatality rate, no cure, and has killed thousands of people. This video will explore the protein structure of the virus, as well as research attempts to develop a vaccine against it. Vaccines function by exposing the body to antigens of a virus, so that the body can develop a large stock of antibodies that specifically recognize the virus's antigens, and will be able to respond quickly and effectively to infection by the true virus. In the case of Ebolavirus, the antigen that the immune system's antibodies respond to most readily is a glycoprotein located on the exterior membrane of the virus. The glycoprotein is a symmetric protein made of 3 identical pieces, called monomers, which bond to each other to form a chalice shape. Each of the 3 pieces of the glycoprotein is made of an inner protein structure called GP1, each cradled by a protein strand called GP2. To generate an anti-Ebolavirus vaccine, researchers use a non-lethal virus called vesicular stomatitis and incorporate the Ebolavirus glycoprotein gene into its genome. The vesicular stomatitis virus can then express the glycoprotein antigen on its surface and be used to make a safe vaccination. The human antibody KZ52 has been found to recognize the glycoprotein, binding simultaneously to the GP1 and GP2 strands. Because the virus uses the glycoprotein to attach to host cells and invade them, the KZ52 antibody can neutralize the virus by preventing the glycoprotein from functioning. In the black box, we see the chemical side chain interactions between the KZ52 antibody and residues on both GP1, represented in green, and GP2, represented in yellow. The residues interact through both van der Waals and hydrogen bonding, with 15 interactions in total, such as this interaction between asparagine on the glycoprotein and tryptophan on the antibody. There are actually several different closely related species within the Ebolavirus family that cause the disease. The most common and deadly virus is Zaire Ebolavirus, but species known as Côte d'Ivoire, Sudan, and Reston have also been identified. Each of these species has a slightly different glycoprotein structure, so a vaccine that introduces an antigen from one species may not protect against another species. The KZ52 antibody, for example, only neutralizes the glycoprotein of Zaire Ebolavirus. This figure compares the glycoprotein peptide sequences of the Zaire, Côte d'Ivoire, Sudan, and Reston Ebolaviruses. The asterisks represent the areas of the protein that interact with the antibody. In these crucial areas, there are several different residues present in different species. These small structural differences make it hard for an antibody that recognizes one species to recognize another, meaning that vaccines that protect against one species may not protect against other species. In 2011, Marzi et al. Conducted a study testing the cross-protective efficacy of vaccines that express glycoproteins for Zaire, Sudan, Reston, and Côte d'Ivoire Ebolavirus. Guinea pigs were inoculated with these vaccines and then exposed to a lethal dose of Zaire Ebolavirus. All of the guinea pigs inoculated with the Zaire Ebolavirus glycoprotein survived exposure to Zaire Ebolavirus, but less than 17% of those inoculated with glycoprotein vaccines from the other Ebolavirus species survived. This data shows that vaccines designed using one species' glycoprotein are not effective at protecting against another species. To develop a vaccine with better cross-species protection, the lab created a recombinant vesicular stomatitis virus that expressed not only the Sudan Ebolavirus glycoprotein, but also another Ebolavirus immunogenic protein, VP40. Only 1 out of 6 guinea pigs administered with 2 doses of this vaccine succumbed to Zaire Ebolavirus, showing that a vaccine with better cross-species protective efficacy can be achieved by creating a recombinant vesicular stomatitis virus that expresses more than one Ebolavirus protein. As we have seen, the existence of different strains of the virus makes it hard to develop a vaccine with high cross-species protection. However, results from the MARSI study are promising and will hopefully lead to the widespread implementation of cross-species Ebola virus vaccination.